Maybe good morning, good afternoon, or evening, depending on where you are joining from. First of all, I'd like to thank the organizers for setting up this workshop. And my name is Mert, and I'm from the University of Groningen. And today I'll share our work on molecular dynamics simulations of the JCVI-syn3A cell envelope. And our group is called the Molecular Dynamics Group, and as the name suggests, we use simulations to capture dynamics of biological systems, from molecules to entire cells. And as you can see here, one example is that this beautiful spherical membrane vesicle. And you can already see that how dense this vesicle is compared to the typical molecular dynamics simulations. And the size is truly huge. And let me start with a bit of background. And molecular dynamics is often described as a computational microscope, and it allows us to watch biomolecules in motion and to connect molecular interactions to biological functions. And in atomistic MD, and every atom is resolved. And this gives us a very high detail, but it also limits the size of the systems and the length of the simulations that we can run. And on the left, you see a simulation of a gangliosite at the atomistic level. It's good, but compared to the coarse-grained simulations, it's computationally demanding. And to reach larger scales, we use coarse-grained simulations. And in coarse graining, groups of atoms are represented as 1 beads, as you can see here. And on the right, you can see the coarse-grained version of the same system, and where the dynamics are still captured, but with much less computational cost. And the specific coarse-grained model we used is called Martini force field, and Martini offers flexible mapping. Typically, it's 4:1, but sometimes you can go with 3:1 or 2:1 when you need more resolution or when chemical specificity is needed. And it's widely validated across biomolecules, like from the lipids to proteins, nucleic acids, carbohydrates, or even polymers. And importantly, Martini supports very large-scale simulations up to the whole organelles, or hopefully entire cells. And I just said hopefully, but actually my colleague Jan is already getting very close to this goal. In our group, under Jan's lead, we've been working step by step towards these whole cell simulations. And the aim of these simulations is to capture the complexity of an entire minimal cell. And of course, to integrate the chromosome, cytoplasmic proteins, the envelope, and all other relevant components into a coherent model. And of course, this is not an easy task. It requires combining many different types of data and also developing new tools to make this visible at this scale. And within this broader project, my own focus is to the envelope of this cell, the minimal cell. And by envelope, I mean the lipid bilayer and the embedded proteins on it. And here you see the Martini model envelope of JCVI-syn3A that we've been able to simulate. Maybe you can see the video, just little moving. And this is about 400 nanometers in diameter, very close to the real size of syn3As and maybe the small ones. And it contains 4,355 proteins embedded in the membrane, which adds up roughly to 0.7 billion coarse-grained beads. It's of course difficult to appreciate the level of detail because it's currently only 100 nanoseconds. But the reason is that running the system is extremely hard and computationally demanding. So we have to proceed carefully as we extend it further. But still, this snapshot gives a sense of the scale we are now able to build and simulate. Okay, how do we build these such large systems? Let me show you the workflow. We start by selecting proteins based on the proteomics data, thanks to our collaborators. And the structures were predicted by using AlphaFold tree. And on this slide, you can see the workflow. We start with the atomistic structures, then we Martinize them to get the coarse-grained structures, and also we embedded them into bilayers to relax the structures and also get the position of the transmembrane region in the membrane. And you see the 94 different types of membrane proteins here. We activate them up to 1 microseconds. And eventually, we integrate them into spherical vesicles. And we use very good tools like JCVI-syn3A and TS2CG. Okay, now let's turn to the lipids. Here you can see the two vesicle hemispheres representing the two different compositions. On the left, this is the validated lipidome of syn3A, a 5-lipid mixture containing high cholesterol, and the other 4 different lipids like sphingomyelin, cardiolipins, POPC, and DOPG. And on the right is the minimal lipidome that's recently published, which consists of only 2 lipids, which is the cholesterol and ether-linked POPC. And both systems are cholesterol-rich, but the 5-lipid system has the charged lipids like cardiolipin and DOPG, which are absent in the simplified model. And we thought that that will be interesting to analyze these systems and maybe collect some data about lipid-protein dynamics from this comparison. And for practical reasons, we choose to simulate the systems at in a diameter of 200 nanometers rather than the full 400 nanometer scale. And this allowed us to run them for longer time scales. And directly we can compare the effects of lipid compositions under these conditions that are still biologically meaningful. Okay, for these systems, spherical integrity is particularly important because we want to make sure that what we analyze is a stable vesicle. And in figure 3 on the left, you see the lipid number versus target radius we want to simulate, and together with the snapshots after the just initial equilibration. And this means if you start with densities that are too high or too low, the vesicles look distorted at the beginning, wrinkled in some cases, or flattened and in others. And different lipidomes has different packing properties. So to avoid wasting computational resources on these for these unstable large systems, we optimize the packing density before production runs. And by tuning the area per lipid, which is the way in TS2CG, you can adjust the density of your vesicle. So we can start from vesicles that are already balanced. And to achieve this, we start with small vesicle simulations and predict the trend that gives us a better initial configuration specific to these compositions. And on the right, you can see that the internal vesicle volumes over the 2.5 microseconds, and starting from the optimized density, the volumes remain stable, and this shows that the geometry preserved over time. So in short, careful packing at the beginning ensures that the vesicle maintain the spherical integrity. Let's turn to the proteins again. Both systems remained stable on the microsecond scale, and here we run them up to 2.5 microseconds. And distinct clustering patterns were observed. In 2 lipid vesicle, clustering progressed faster, and in A you can see the average size versus time. And in B, a snapshot of the largest cluster observed in these systems. Here you see in red. But we don't know when it's fully converged, both systems may reach similar cluster size. But to get a better understanding on the time scales, we run smaller analogous flat bilayer simulations for these 2 systems. And here you see the initial system, similar to both systems. And you also see the final snapshots after 20 microseconds. And we observe the same trend, and which means that for us, 2 lipid system cluster faster initially. But however, in my latest runs, proteins in the 5 lipid system also reached the similar cluster sizes. But you can see that in both systems, the 2 lipid system progress faster clustering. Now I would like to show you a short movie from the 5 lipid system. Can you see the movie? Moving? Yeah. Okay.
Yes.
Nice. We originally prepared this video for the opening of the Feringa building on our campus, so it's a bit of a showcase. Here I zoom into the one of the protein clusters, and you can actually see a protease coming into contact with ATP synthase. Here, maybe you can see we are zooming out the protein cluster formation. But I should emphasize that the protein positions are completely random in this setup, so this does not carry any biological meaning yet. But still, it gives a sense of these kind of interactions we can visualize. by using special tools like VMD. And perhaps maybe in the future we can aim for studies where such interactions are modeled more realistically at this scale. We have the data for it. Now let's look at the lipids again, and in particularly how they arrange around the proteins. And in figure 4, you see the time evolution of the lipid arrangement and depletion close to the proteins. And some lipid species, like cardiolipin, show clear accumulation around proteins, while others excluded around the proteins, like sphingomyelins, which is expected. And on the right, you can see the hotspots that shows the cardiolipin arrangement on the 5-lipid vesicle. And to illustrate this more directly, figure 5 in A, you see the initial frame of the 5-lipid one. And in here, cardiolipins within 1 nanometer of the protein positions are marked on red. And on the right, you see the final frame, and you see that the density of cardiolipins around the proteins has strongly increased. And finally, in the close-up of ATP synthase, you can clearly see that cardiolipins are clustered around the ATP synthase. And in another study, I turned to the protein-free vesicles, which we use to isolate the role of cholesterol. And at around 60% cholesterol, which is also the native level of syn3A, the vesicle remains stable, which also expected. And cholesterol intercalates in these systems. Cholesterol intercalates between neighboring lipids, condensing then ordering the bilayer. And also, when we systematically varied cholesterol levels, cholesterol concentrations on small flat bilayers, we saw that the classic sterol effects, with more cholesterol, the bilayer become thicker, the areal per lipid decreased, and also the lipid order increased. And we also found that bending rigidity increased and diffusion of the lipids decreased. You see here. Just that, sorry, something? No, okay. And we also see that the properties peaked at after 60% cholesterol for all the analysis we done. And this suggests that syn3A membranes operate close to the sterol solubility limit, where the membrane is maximally stiff, but it's still functional. And altogether, I can say that these results highlight this cholesterol still is the key determinant in these membranes of these minimal cells. And to wrap up, with Martini, we can now simulate these systems at the scale of entire cell envelopes, and not only for synthetic cell. Also, they also opened for studying membrane biology at various scales more generally. And third, our analyses are still going, and I've shown you clustering, lipid arrangement, and cholesterol effects, but there's much more to come. Those are only the initial results. We plan to explore more. And some of the candidates are lipid flow streamlines, and also the simulations of membrane responses to osmotic shock by using tools like a shocker. And finally, I would like to thank all of you for your attention, and also my colleagues and collaborators for their contributions to this work. Thank you.
That was terrific, Mert. Questions? I'll start. So it does seem like, you know, we need to get some cells in you guys' hands.
Yeah.
And given the work that James Sayens has shown that you can alter the composition of lipids in the cell growth media, and the cells are governed by whatever you choose to give them, it would seem like that you have a great system available to evaluate how effectively your models are fit with actual experimental data. Ingguang,
yes.
You have a question?
Very cool research. So just a quick question about the number of membrane proteins you put into the lipid. It looks like a little bit low for me. How much is the percentage of like transmembrane proteins occupied these lipid surface area? And are you considering increase that number?
Yeah, for the small ones, we reached the 25%. 25%, but the larger one is getting better, 40% lipid to protein ratio on surface.